All right, good day, everybody. Good day, everyone. All right, good day, everyone.
The
Yes, we are very much delighted to have each and every one of us on this call. And all right, we will be starting the graduation ceremony shortly. I will just need more people to join this call. So we just need more people to join this call, and we will start the graduation ceremony. While we wait, you can share the YouTube link with your friends, your families, your colleagues, so they can join to celebrate with you and also to see how much you have done and how much progress you have made. Because I am certain that you have definitely improved, you have definitely upskilled. You are not just the person you used to be 3 months ago. So you can do well to share the links with your friends, with your colleagues, with your family, and for other classmates. I guess I can use the word classmates. Yes, you can also try to reach out to your other interns, your other colleagues that you work together during this research internship, so they can also join and um yeah, so they can also join the graduation ceremony. So shortly, we'll start shortly, but while we wait, we can also do that. We just need to be more, just need more people on the group, on the call rather. Thank you. I guess you can continue now. So, good day once again. And like I said earlier, we're very happy to have you all on this call. We sincerely celebrate with you as you finish your internship, as you conclude your internship for this cohort. And we have seen your hard work, your commitment, your dedication so far to your work, to not just learning, but to becoming a better person, um to becoming better even in your research, in your field of study. And we really celebrate you and commend you for your job well done for the times. We know it wasn't really easy for many of us, but we celebrate your tenacious spirit and your commitment to your work. So without further ado, I will just commence the ceremony. All right, so I would just be talking briefly about Genomark Institute, of which I know many of us know already, but for the sake of those that do not know, and prior to this time, I know I asked us to share the YouTube link with our colleagues and friends. So it's very possible we have people that do not even know what Genomark is at all. And please, while we are still on this call, we can still keep sharing the link with with everyone we know should be on this call. It's not just for the grad ones, but also for as many that also want to improve in their field. So as much as it's your ceremony, as much as it's your ceremony, it will also be nice that you also invite more people to celebrate with you. All right, so Genomic Institute is committed to advancing research and education in genomics. And that's why we have various programs to help everyone understand, help people understand genomics and how it relates to their field. All right, so I know this is not our first time of having to of hearing anything about genomics, and yes, Genomic Institute. And like I said earlier, it's majorly for those that do not know, and also if we ourselves want to talk about Genomark, we would definitely know and have something to say. All right, so I also bring greetings and congratulatory message from the director of Genomark Institute, the person of Mr. Oluwaseun Agbola. He sent a sincere greetings and congratulatory message to you all, and commending you for your job well done so far. All right, so we will be going directly into the presentation for each group. Yes, we'll be going into the presentation for each group. I know we have the representative for each group on this call: cancer genomics, anti-cancer, infectious diseases, and other groups. But if your rep is not on the call, if your representative is not on the call, you can please reach out to them already so they can join this call. So I'll be going straight to the presentation. Do we have the representative for cancer genomics on this call?
Yes, ma'am. We are here.
All right, now you can go ahead with your presentation now. And please let's take note of this. We have 5 minutes each for presentation. Thank you.
Okay. Can everyone see my screen?
Yes, ma'am. I see your screen now.
Okay. So, good morning, good afternoon, good evening, forever you're in the world. Um, this is a presentation by Cancer Genomic Group, and our research topic was genomic profiling and variant analysis of colon cancer tumor. investigating mutational signature and their impact on tumor progression and clinical outcome. So colon cancer, as we do call it, mostly CRC, is a type of cancer that affects the colon, which is the largest intestine. And according to WHO, it is the third most common cancer worldwide. And it's also recognized as uh, contributing to 10% of all the cancer cases in the whole world, and also second leading cause of cancer-related death worldwide. Um, Also, the development of CRC is a kind of a cascade of coordinated mutational event in the signaling pathway. Various genes that associate the signaling pathway have been reported to be dysregulated. Example, the EGFR, the MEP kinase, and the RAS, and the PI3K. And NCBI houses most of these data sets of both RNA-seq analysis and the WGS. However, majority of these data sets remains as a raw sequencing grid, which means it limits like a comprehensive downstream interpretation of what these data sets mean clinical-wise. So our goal was to conduct genome profiling and variant analysis of CRC data sets with the objective of investigating the mutational signature and their impact on tumor progression. Our specific objective was to compare the sequence read from the NCBI dataset database. We compare them to the reference genome and perform variant calling. And we also construct variant profile for each sample, classifying the polymorphism type and the protein effect. And then at the end, we compare shared versus unique mutational event across all samples. So our hypothesis was that WGS of colon cancer would review unique and shared mutational events, which will reflect canonical pathways and also tumor heterogeneity. So this is our workflow. First of all, we acquired our data set and we processed them from the NCBI SRA. We had about 10 data sets of different variant sizes, and then we moved straight to the quality control and genome assembly using SPADE assembler. And then after that, we mapped to reference. The reference genome we used was the GRCH38B14, which is the chromosome 5, and then we used the genome software for that. We also did genome annotation using the same software, and then we did variant calling. After variant calling, we merged all the data samples and then moved on to Tableau for data processing. After data processing, we visualized the data using Excel, R, and ClassVS. So here is our finding. So in figure one shows the percentage contribution of variants per sample. So we had a total of 251 variants, and on the y-axis is the percentage each of the sample contributed. Like the highest was sample 7, and the least was sample 6 and sample 2. Okay, so this shows variation among all samples, and this could be because of the different sizes of the sample size of cohort. And then on figure 2, here we show the total gene. The total gene observed was 5,522, and this does not match the total variance of 251. This is simply because most of these genes were like literally reoccurring, like their frequent were so much. And out of the whole gene, the 10 top frequent genes we observed was the BRAD9, the DN, and like the DNA, JC21, and the rest. And several of these genes are linked to transcriptional regulation, metabolism, and cancer-related signaling pathway. And then here is also a figure for the type of polymorphism. For figure 3, SNP accounted the majority of the variant, with transition being more common. We had indels to be less frequent, but they were present across all samples. And then for the protein effect, it was non-synonymous substitution was majorly what contributed to the protein effect. And we also went further to check the unique versus shared mutational event. So we had a total of 424 events, and 38 were shared among all. So these are like variants that occurred in all samples and variants that are unique to each sample. So we had 90.3% of variants that were unique to each sample, and then 9.7 that were shared amongst the samples. However, a breakdown of the 9.7% was that no particular variant was shared across all 10 samples. Rather, you could see in figure 6, two samples shared 24 variants, three samples shared 8 variants. So there is no single variant that was shared across the whole sample. And this could maybe point towards um, tumor heterogeneity, and maybe precision could be helpful for precision medicine. So our discussion. The goal of the research was to investigate the signature, mutational signature in CRC. And based on what we saw on figure 1, the huge data we had, the huge variant we had suggested mutation load, and this could reflect genomic instability. And genomic instability is central to CRC. This is what every literature know, everybody know. And this can be helpful in prognosis because usually high mutation is a first signal of aggressiveness. Okay. And also, among the recurring genes observed as seen in figure 2, BRAD9 and SDHA had the highest frequency. This pins down their role in tumor progression. And BRD9 is unique for chromatin modeling, and SDHA was like in the mitochondria. So they both reflect epigenetic dysregulation, while SDHS reflects mitochondrial dysfunction. However, the surprising thing we discovered was that none of the major drivers of CRC was observed in our sample. And this might be because of our sample size. At least we need like over 100 samples if we would review them. So probably because we had a small sample size. But the most frequent gene observed could be a therapeutically promising target that like for clinical treatment. Again, the abundance of SNP observed indicates point mutation as the primary driver of oncogene, and which is for like what is the norm. And then consistent presence of the intel, even though it was less frequent, may reflect underlying genomic instability, as I said before. And it's also a sign of high mutational burden. And also, the non-synonymous substitution we observed indicates that majority of the mutation are likely to alter protein structure and function, like oncogene activation and the tumor inactivation of tumor suppressors. And predominance of this non-synonymous substitution in our data set may highlight their central role in tumor progression. Finally, no single mutational event was shared across all samples. This could suggest heterogeneity and maybe possibly that each of the tumor followed its own evolutionary trajectory as observed in CRC. And this also suggests that personalized medicine is crucial in CRC. In conclusion, based on our studies, CRC displayed a complex mutational landscape dominated by SNP and maybe a little bit of gene indels. And most of these mutations are non-synonymous, indicating strong potential to alter protein function and drive tumor progression. While we did not observe the major genes in the CRC signaling pathway, emerging alteration in genes like BRAD9 and SDHA highlights additional role of epigenetic and metabolic dysregulation. And also the high heterogeneity in our sample here is also important for precision medicine. Here is our selected references and acknowledgement. We would like to thank, first of all, the editorial team for making this a reality. Without them, this presentation wouldn't have been a reality. We also thank our classmates for helping finding answers to individual questions for the collaborative effort to ensure that we carry on this project. We also thank our mentor, Miss Olabi Blessing. You're such a lovely woman, and we want to thank you for your invaluable guidance and your patience and your support throughout this project. We wish to thank Genemark Institute for their initiative in facilitating this research internship. And this work would not have been possible without the contribution of every individual in the organization. Finally, we say thanks to God for the zeal and the strength to complete this course. Thank you.
All right, well done, Ma. Well done to the whole Cancer Genomics group. That was indeed a wonderful presentation and was really, really insightful. Thank you very much, Ma. And we also celebrate the whole team. And we indeed see that you had put in much work and effort and intentionality into learning and also into this your presentation. Well done. All right. So I'll be calling on the next team, the next group to present the infectious diseases group. I don't know if they are ready. Infectious diseases. All right. I think the rep is on the call here. Okay. Can we have the rep for the anti-cancer group? Anti-cancer. Can we have the rep for anti-cancer group? Okay. All right. The infectious disease group representative. Are you on the call? Mr. Victor, please can we have a presentation for infectious diseases?
Okay,
Yes, sir. And sir, please, you have 5 minutes. Thank you. All right, we can't hear you, sir, and we can't see your screen also. Mr. Victor.
I can hear you now.
Yes, you have the floor. Please go ahead with your presentation.
So please, can everybody see my screen?
Not yet, sir. We can't see your screen, sir.
Okay. So can everybody see this?
Yes, sir, we can see your screen.
A mix of multi-drug resistant pathogens in healthcare.
All right, sir, we can't see what your slide is not showing.
Okay.
All right, while we are still waiting for him to come up with presentation, I would want us to take note of the following announcement, or this announcement rather. Your certificate will be made available latest by Monday. So you would have access to getting your certificate on Monday. Starting from Monday, you would have access to your certificate. And also, for as many that will be needing a recommendation letter, a number will be sent to you, you send across to each group. to reach out to, you would reach out to that person to get your recommendation letter, and for whatever you need it for, you would reach out to that person, stating your reason, or you're stating your reason for requesting for the recommendation letter, and it will be addressed. So we won't be issuing the recommendation letter to you. We will be directed to the person you'll be getting the recommendation letter from, and your certificate will be made available starting from Monday. So starting from Monday, you can begin to get your certificate. All right. I think he's not on the call again. Mr. Victor, are you on the call? Are you still on the call?
Listen, I want to know whether you are hearing me. Confirm whether you are hearing me. I'm from the cancer group.
All right, sir. All
I've
right.
been struggling with my mic.
All right. We can hear you, sir. I guess we would have to come back to Mr. Victor for the infectious diseases group. So, Dr. Emmanuel, please, you can go ahead with your presentation for the anti-cancer group.
It's okay.
And please, you have 5 minutes, sir.
Are you seeing my screen?
Yes, sir, we can see your screen and we can hear you audibly.
Okay. Thank you very much. Okay, thank you very much. This is anti-cancer group. This is what you work on. Anti-cancer efficacy of streptomycin-derived therapeutic peptide against lungs, breast, and prostate cancer model. And these are actually our mentor, Aditola Adisona, and these are actually the students that work on the group. And this is the background. Of course, you know that cancer is a leading cause of death globally. Resistance, and one of the challenges we have in the cancer therapy is the resistance and the toxicity limit in the current therapies we have. And as a result of that, there's a need to get an alternative that will actually help out. And so the peptides from streptomycin species should promise as anti-cancer agents. So that's why we work on that. So the machine learning accelerates the discovery and optimization. Now, this is the aim of this study. Are you hearing me? So this is to investigate the anti-cancer efficacy of streptomycin-derived therapeutic peptide against lungs, breast, and prostate cancer models, while using machine learning tools to predict, optimize, and validate its properties. These are objective: to isolate, characterize, and validate the therapeutic peptide from this streptomycin onboards, and then to apply the machine learning for predictive models.
Your slide is not moving.
Hello? It's not moving?
Yes, it's not moving.
Wow.
You can put it in a slideshow.
I put it in a slideshow and it was moving on my own side.
All right, it's not moving. You can try to put it in a slideshow again.
I should try to put it in a slideshow again. Are you seeing it now? Is it moving now?
No, sir.
Is it moving now?
No, it's not. It's not moving,
Let
sir.
me return it to remove it from slideshow. Probably. Is it moving now? Hello?
It's
Yes.
not moving. It's not moving.
Okay. It's moving already, sir. Your slide is moving already. You can continue, sir. Dr. Emmanuel, you can continue with your presentation. We can't hear you again, sir. All right, Dr. Emmanuel, if it's not moving with the slideshow, I think you should just continue and move it manually, like yourself. So you can continue. We don't have much time. Can you hear me, Dr. Emmanuel? Dr. Emmanuel, please, we can't hear you. Your mic is either muted or there is an issue, but we can't hear you at all.
Hello, are you hearing me now?
Yes, we can hear you now.
Okay. Sorry for that. Okay, I'm still in the objective, and this is to compare the peptide efficacy and mechanism across the lungs, breast, and prostate cancer. Then the methodology, do sample collections. This
Sorry,
study
is
is
it possible
utilized.
to start from the beginning again?
Hello?
Is it possible to start from beginning again, please?
But you see the slide now?
Yes, very well.
Okay, so let me start from the beginning. So this is the anti-cancer efficacy of streptomycin derived therapeutic peptide against lungs, breast, prostate cancer model. Is anti-cancer group in this internship. These are the names of the people and the name of our mentor. So I say the background is that the leading cause of death globally is this cancer. And one of the challenge we have is the resistance and the toxicity limit that limit the current therapies we are having in anti-cancer. So beta from streptomycin species show a promise as an anti-cancer. So there's a need for an alternative since there's a resistance in the current ones we have. And we use machine learning to accelerate the discovery and optimization. These are actually the to investigate.
Emmanuel,
Yes.
can you please move your slide, like yourself? No, it's
Hello?
static. Yes, sir. Can you move your slide? It's
I'm
static.
using the slide.
It's static. It's not moving again.
Oh my God.
You don't have to like stop
My
presenting.
slide is turning here and I don't know why it's not showing the screen there.
All right, no problem. You can just continue.
Okay. So I said the aim is to investigate anti-cancer efficacy of streptomycin-derived therapeutic peptide against lungs, breast, prostate cancer model while using machine learning tools to predict, optimize, and validate its properties. So our objective is to isolate, characterize, and validate the therapeutic peptide from streptomycin onboards, and also to apply machine learning for predictive modeling and optimization of peptides, and to do the compare those peptide efficacy and their mechanism against lungs, breast, and prostate cancer. And then we do a collection of sample. This study is to utilize the data sample from sequencing of streptomycin onboards. All raw data sample were collected and freely available in NCBI. with the projection access number. And then these are general sequencing properties, which we collect. We have by a project, we have by a sample, and then we have our SRA. Each of us work on 2 samples each, which are around 6 or 7. So that will be a total of 14 samples. And we have the size of those sequence, and then we see the isolation source. For this one, the isolation source is the drainless lake, which is a salt water. And then with the year of the isolation. Then what is our methodology? We do characterization of a secondary metabolite gene cluster. So it's going to mycin onboard sample isolated from this lake.
Slide is not even moving.
You can't hear me again?
No, sir. Please continue, sir.
The antimartial was utilized a software pipe access via the website, and this is actually the metabolite. If actually you are seeing it, I will show you the core biosynthetic gene and the regulatory gene, and that is actually our focus. And when we isolate that, and then we do the prediction of it, and to get the secondary metabolite, the encoding gene, the putative functional listing, which is the biosynthetic, and we also have the protein and the location, the amino acid length and the score and E value, and the sequence there also is a bit we are not seeing it. We have the sequence which we isolate. Then we use machine learning for predictive modeling and the optimization of the peptide. The Cap3 tools renowned for its advanced machine learning algorithm was employed to systematically identify this antimicrobial peptide derived from biosynthetic gene, thereby enhancing the accuracy of peptide selection for downstream analysis. Utilizing the suite of machine learning models. What are those machine learning models? We use support vector machine, we use random forest, and discriminant analysis, and artificial neural network. These are what we use to evaluate this antimicrobial, and this is what we have, the predicted antimicrobial peptide within the peptides. And then we go on to see those ones that are mutant, and then see their probability. And with all the machine learning, they all predicted that they are anti-cancer peptides. And we have the 4 major ones that stand out of all the peptides. And we go on to characterize and do selection of this therapeutic. And so the selection of peptide, this potential anti-cancer activity was conducted through systematic evaluation of key physicochemical properties. And then we talk about other things. And these are the 4 anti-cancer therapeutic peptide. Out of the month we did, we select the 4 major anti-cancer peptide for further studies. And we have them, we bed them, we have PD101, that 10116, and other people have their own. But out of this, we have 106, 3, PD105, and PD11 are the one that show most. The other ones show the same, but this one is the top, the 4 top. So we have assessment of this potential peptide selection. Like I said, from the total of the peptide analyzed, the study, we have the 4 promising candidates for anti-cancer activity. And the source, we have the source of this peptide from 1 acid transferase with beta-rollatone as secondary metabolite. Other ones, the same thing. And then we go ahead to talk about the effect of this once we discover in each of the cancer we mentioned. Now, let's talk about the peptide efficacy and mechanism against the lung cancer. Then we have the first one, which is the one we have mentioned before, which is the peptide number of 106.
Dr. Emmanuel, please, can you begin to round up?
Hello?
I said, can you begin to round up, sir?
Okay. So I'm just talking about the effect now on the lungs. We have all of them. Just in conclusion, we talk about it in the lungs, we talk about it in the breast, and we talk about it in the prostate cancer. If you have seen my slide, we have seen the effect in all this that the cancer in both breast lungs. So what is our finding? So the PETA show a selective cytotoxicity in lungs, breast, and prostate model. Machine learning predicted enhanced stability and reduced toxicity. And comparative testing show hypothesis induction with pathway variation. What is the contribution to knowledge in this research? Is a novel research, it's novel insight into streptomycin umbus as a source of anti-cancer peptides. That means that those bacteria have anti-cancer peptides in all those areas that can help, and also demonstrated machine learning role in peptide drug discovery. Framework for comparing the peptide activity across the solid model. What is the limitation? The limitation is that we actually need the in vivo validation on this. And there's also a need to test the efficacy in animal model. Then we can also expand the machine model with larger peptide data sets. In conclusion, the streptomycin umbus peptide show broad spectrum anti-cancer potential. So machine learning also enhanced prediction, optimization, supporting ISO as a candidate for peptide-based cancer therapy. These are reference. So I want to thank all the group members and our mentors for their guidance. Thank you very much.
All right, so well done, sir. Thank you very much for your rich presentation. Yeah, though the slide was not moving, but indeed we see that you had put in much effort, much time, much dedication to your work, to learning, to doing better, to coming up with all of these that you have presented. Well done to you, well done to your whole team. And once again, we congratulate you for how far you have come with your work. All right, so the next group that will be presenting is infectious diseases. All right, can we have the group rep to speak? All right. Okay, Mr. Innocent is asking, are you asking for us to share the slide for anti-cancer genomics?
Yes, ma, since we are not able to see what he was presenting.
All right, so we'll see to that. We'll see to, you are in what group, sir? Because I don't know if other groups are interested in seeing the slide, but you're in what group, so we can send it to you, or would better still, we'll send it to all the groups. Or all right, okay. So we'll share the anti-cancer group slide to your group, so we can see all that was spoken about. All right, thank you very much. Okay, so we have the rep for infectious diseases already. So you can go ahead, Mr. Victor.
Okay, so good afternoon, everybody.
That's nice.
I hope everyone can see my screen now.
Yes, we can see your screen.
All right. So my name is Onekena Victor, and my research focus is on infectious diseases with a focus on antimicrobial resistance. My research topic is comparative genomics of multi-drug resistant pathogens in healthcare, and my focus is on Mycobacterium tuberculosis. Okay, so this is my table of contents. Can we all see it?
Yes, sir, we can see it.
All right, so this is my table of contents, and we go to the introduction. Now, tuberculosis is one of the top 10 causes of death worldwide, according to WHO, with nearly 10 million new cases announced annually. Now, the drug-resistant forms of tuberculosis present public health emergency to the whole world. And we talk about genomics. Genomics is the study of the complete DNA of an organism. And when we are talking about the complete DNA of an organism, we are talking about both the coding and the non-coding region of DNA of the organism. And advances in sequencing technologies have made it possible to discover mutations that drive drug resistance in Mycobacterium tuberculosis, helping in diagnosis, treatment, and surveillance. We talk about comparative genomics, and when we are talking of comparative genomics, from the word comparative, we are comparing genomes of different strains or species. So we have types of antimicrobial resistance in tuberculosis. That's when we are dealing with tuberculosis. We have the drug susceptible TB, where the strains respond to all first line drugs, and the standard 6 month regimen is very effective. We have the monoresistant TB, where the strain is resistant to one of the first line drugs. We have polyresistant TB, where the strain is resistant to more than one of the first line drugs. And we also have multi-drug resistant TB, which is just like the polyresistant TB. So we have other types of antimicrobial resistance in tuberculosis, like rifampicin resistant TB, extensively drug resistant TB, and resistance to newer drugs, which present emerging concern to healthcare globally. So my research aim and objectives. For my research aim, the aim of this research is to understand the genetic mechanisms underlying multi-drug resistant pathogens and comparing the strains from clinical sources. My research objective is to identify and compare genetic determinants of multi-drug resistance and to understand the mechanisms and the spread of this resistance. My methodology. This is a flowchart showing the detailed steps for every method that was employed in this study. The first is data retrieval, where we got our data from online repository such as NCBI pathogen database. And before that, we all know that the sample has to come from the clinical source where it will get sequenced, and then the information from the sequencing will be transferred to an online repository. Now, this led to my group selecting samples H190007 and H190008. Then the second step is quality control and genome assembly, where we made use of raw GS data. We use the BVBRC genome assembly pipeline, employing assemblers like Unicycler, FlyE, post-processing tools like SAM tools, and assembly quality assessment, QAST. Now my results. For my results, this is the result from my function annotation, which shows the strain, and under the strain we have plasmid. The strains do not have a plasmid, but the genome length of the strain can be seen to be 4.3 million plus to 4.4 million plus, and the contigs 93 and 2. Also, we have the contigs N50 to be 129,968 and 4,445,944. So the contigs and the contigs N50 are just referring to the fragment of the genome which is being pieced together. So the smaller the contigs and the larger the contig N50, the better the quality of the genome assembly. Why the larger the contigs and the smaller the contigs N50, the poorer the quality of the genome assembly reports. So for the G plus C content, guanine plus cytosine content, we have 65.58 and 65.59 respectively. So there are other information that are being displayed from here, such as the coding sequences, where we have 4,290 and 5,295 respectively, functional protein, hypothetical protein, RNA genes, CRISPR repeats, and genes with PGFAM. So for the CRISPR repeats, we see that we have 53,000 for H190007 and 177 for H190008. Now this confirms that this strain H190008 is able to develop better immune response to evade any infection by foreign bodies like phages. Okay, then we go to so for antimicrobial resistance results, From here we can see the strains. So we'll be using only just one strain, and from here we can see the antimicrobial gene, the product of the gene, the classification, the antibiotic class, and the antibiotics. So let me use the first colon for the first. Okay, let me use the first row. For the first row, we have H1907 with the FAB gene, which codes for the 3-hydro 3-hydroxy acyl CoA dehydrogenase, and sorry, the antibiotic class that targets this product is triclosans. Then we can also see from here also H1907 RPSL. We can see the product. We can see okay, sorry. For the H1907, the antibiotic class is also triclosan, and the antibiotics is also triclosan. So this shows that this table is just showing us the strain, the antibiotic resistant gene in that strain, the product of such gene, and the class of the antibiotic that targets such gene, or let's just say the drug to which that gene is conferring resistance to. Then we move to the AMR data visualization. From the data visualization results, we can also see from here, we can see this heat map. This heat map is showing, okay, on this row, on the y-axis, we have the antibiotics, the antibiotic class. While on the x-axis, we have the genes that are present in that strain. So for each box that you see with a green color, that is trying to show you that this particular strain has this gene which is conferring resistance to a particular antibiotic. For example, now if we look at this triclosan, when we trace it down, we can see that it is the gene that is conferring resistance to triclosan is the FAB gene, which is what we can also see here on this antibiotic and antimicrobial resistance results on the table here. So the FAB gene is what is conferring resistance to triclosans. Now, my conclusion: high quality genome assemblies of Mycobacterium tuberculosis isolates were obtained using the BVBRC pipeline with tools such as Unicycler, FlyE, SAM2, Pylon, and Quast. Functional genome annotation using RAST-TK and gene ontology revealed thousands of coding sequences, functional proteins, and RNA genes, providing insight into the metabolic and resistance pathway. The AMR heat map highlighted strong resistance gene signals against isoniazid, rifampicin, aminoglycosides, and peptide antibiotics in specific strains. These findings mirror the clinical progression from multiple drug resistant tuberculosis to pre-extensive drug resistance or extensive drug resistant tuberculosis, reinforcing the urgent threat of drug resistant tuberculosis. So overall, integrating genome assembly, functional notation, and AMR profiling offers a robust framework for tracking resistance evolution and guiding strategies against multi-drug resistant tuberculosis. So these are my references for which the relevant literatures were gotten from. And okay, I would like to thank Genomark Hub for sponsoring this training. I would also like to thank my mentor for putting us through in areas where we are lacking, and also my teammates for their efforts, their combined efforts towards this. So thank you for listening.
All right. Thank you very much, Mr. Victor, for your presentation. And thank you to the whole infectious disease team. Well done for your work so far. Well done for your presentation. Your presentation was indeed an amazing one. Thank you very much. All right, so I think we're supposed to have the plant genomics group present now. I don't know if they're on the call yet, but if not, we'll be having a short orientation for the new cohort that will be starting very soon. All right, so we'll be having a short orientation for the new cohort that will be starting very soon. And after that, I think we should have the plant genomics group ready for their own presentation also. All right, so shortly before I hand over to Mr. Francis, who will be taking the orientation session, I would also want to let us know that for each group, we should start writing our research paper. We will be having a month extension to finish up our research papers. So everyone has to, yes, you've worked together well so far, but we need to even do much more and let all hands be on deck to be able to start writing research paper and finish up within the space of 1 month. So for as many that might have not completed their works, please let's go back to finishing up our works quickly and also commencing the writing of our research paper. After today, we have a month extension to be able to finish up our research paper. Thank you very much. So I would hand over to Mr. Francis now to take the orientation session.
Good afternoon, good day, everyone. And I want to congratulate you one more time for the successful completion of your internship with the gentleman. This is a great milestone, and we are proud to see your work done so far. Though the group that presented, they are not the full list of the number of interns that we had for this course. They are just selected ones, and you can see from their presentation that we have indeed gone through the process of them, and then we are all in the working process. So I will, for this session, I'll be privileged to not just congratulating the outgoing cohort, but also welcoming the new cohort, which is the 3B, and getting them ready for their internship program. Without taking much of a time, I would like to share my screen, and then so that we can all go through the orientation. Can you all see my screen? Okay, uh, so we all know that Genomark is one of the bioinformatic company here in Nigeria that have impacts all over the world, not just in Nigeria, but all over the world. Our work span through various sectors like the healthcare sector, the agricultural sector, the biological sector, the tech sector. As you all know that bioinformatics is a biotech thing. It's not just it takes computer and then biology and combine it together to make computational biology. And then we know that the skill of bioinformatics is an evolving skill. It's an evolving skill as the skill of as tech itself is not stagnant, it is always evolving. So we at Genomat, we are also evolving, and we are here to serve you better, to make sure that the necessary human resources and other resources needed for you to be successful and to gain the right knowledge is available for you. So I want to welcome you all, and I want to get you set for the next course. Now, why did we start this internship program? The main purpose of this internship is to equip people to make it available to all of us. So it's primarily a social impact, and it's not just uh, um, a form of revenue. Though we collect a commitment fee, but the commitment fee is way lower than what you get with the same course when you go to other institutions or to other organizations um, to take this internship. So this internship is a virtual internship. It's a virtual program. And what that means that it is a virtual program, I mean that it is something that you can do at your leisure. You don't have to be on ground here at our headquarters for you to be able to take the courses. The courses are already pre-arranged, they're ready to record it, and you can access them wherever you are, anywhere in the world, at your own convenience within the time range, which is 8 to 12 weeks. So it is virtual, and we have heard of many programs that it will be virtual with less monitoring. But our program here is not just virtual, it is engaging, and we can monitor your progress. We have put in place many structures that allows us to monitor your progress while you are gaining knowledge at the same time. So it is a virtual program. It is a beginner friendly. Sometimes you start a program with zeal, with excitement, but when you begin the program, along the way you lose interest because it is too complex. Even me, when I started this bioinformatics, I did not know anything about bioinformatics. The only thing I know is just to go online, read about it, that's all. But when I was able to come to Genomark, I was able to go through the same program that you are going, and I was able to gain the basic knowledge of bioinformatics. So either you are in the area of tech before, either you are in the area of biology before or not, or you know about bioinformatics or not, you can take our program, you can take our courses, and you will get it right from scratch, even to the professional level. So it is a beginner-friendly program, the beginner-friendly training that will help you to skyrocket your career journey in the area of bioinformatics. So what are those things that you are getting? Okay, so what are you getting for this program? So these are some of the things that we can get on this program. But before I go there, uh, Genomark is a well-recognized company anywhere you get to, both in Nigeria, in US, in England, India, China, So our influence is not just limited to this place. So you can search online and you can learn more about this company. So you have a leverage as an intern here to get more opportunities. So anyway, if you go anywhere and you just they see the name Genomark maybe on your certificate, or you present yourself as an intern of Genomark, there is a level of recognition that you get out there when you hold our certificate. So these are some of the things that you get to gain from our course. So you have expert mentorship. We have some people, doctors, master students, undergraduate students, even IT students who have taken our courses, and today they are doing well in their areas of expertise in their career. So we have a whole lot of network of people, of scientists, of career, of professionals, so of professionals exactly, who are taking our courses and who we have developed a wide range of network along the years. So you have expert mentorship with weekly live session, which is worth $500. So our mentors, you are not just giving access to our course on our website. Weekly, we hold live session with you where we get to guide you as you are learning, as you are taking our courses. So these expert mentors guide you anywhere you are not getting it. They are there to explain to you better. They are there to guide you so that the journey of bioinformatics is seamless here at Genomark. So we have trainings and developments you need to get opportunity in your career. I said that Genomark is not just a recognized country here in Nigeria, it is also recognized in many other places of the globe. So when you get our training, you do not just get a localized experience. Because of the experience of our mentors, they tend to expose you to and train you so that you can get opportunities in your career in all of this world. So you have access. One of the things that makes Genomark to be different from all other bioinformatics company in Nigeria, though we work in synergy, is that we have a robust website that you can access many. If I can even show you the overview of the website, it's called the Omis board. Should I do that? Let me just try and show you the overview. If I have, I want to. I see my screen. I see my screen. Huh? Okay,
Yes, it's showing.
Okay, so I will just give me some time. I want to share my own screen. So I'm trying to go to our board to show you an overview for those who do not have idea of what it is. So just give me some time. Are we together? So are we together, please? Are we still together?
Yes, we are.
Okay. So I'm now getting to this board. I will see if I have access. So this is our business program. So you can see on the screen. So we have for each course we have the cancer genome review, Parkinson research, the colorectal research, the prostate cancer research, human microbiome, as you can see. So we have a whole lot of database of classes that of your choice that you can go through. So that is one of the things that make Genoma different from others, and your experience will be very seamless and engaging. So I'm back to my slide. So that is the only spot, and it's worth 1,000, even more, if not more, 1,000. So the only spot is where you have simplified research tools, tags, and submission. As an intern, for your experience to be seamless, we have some we have a platform where you submit assignments, a platform where you can mark your attendance, and you have some research tools like books on bioinformatics, samples of researches or paper that we have published. So if you have you need some books on each topic, on each course, on your specified area that you want to go for. You can download this book. You can download the PDF file of your course for that week. So what we are offering is more than what you get to do. So we have skill advancement that keep you at top wherever you find yourself. So so I've talked about the bioinformatics tools, and one very important thing that we stand to gain in this research, in this intern, is for each course, for each group, there is each intern, or let me say each group, work on a research paper based on what you have done, because this is a hands-on research, is a practical research. You are working on real-life sample that are already on various database like ACBI, etc. So when you get those samples and you analyze them, it is a new research and your findings are new. So you have opportunity to do what? To compile your results and to publish them and to carry, and you have full ownership, full rights to that publication, because we help you in publishing those papers. So I've said that it's very busy, beginner friendly course. So we have some other bonuses that you can gain. You will gain a recommendation letter. You gain certificate. Certificate is always open to you. Connection with professionals all over the world. I've said that, and structured learning and graduation and global presentation. This presentation that we're making today is on YouTube. Many of our partners, many of our students, many professionals, we can go there and watch it again and see what we have done. So you have a whole lot of exposure. As I've said, this is some of our program, as I show you on the always board. We have antibacterial, anti-cancer, antifungal, epidemiology. So for those who are interested in epidemiology, human microbiome, and etc., infectious disease, AMR, infectious disease, relevance factor, plant genomics, viral genomics, etc. So you have opportunity to make your choice. So now, I said that the main reason for this program is impact, and for you to be able to secure your slot in this program, you have to we are charging a $30 commitment fee, which is very, very low to what we are offering, and we have limited space. So anyone that is interested, or you have any friend that you think need to know about this bioinformatics, between now and next week, the application for this cohort will close. So we have limited space, and you can always reach out either to me or to your coordinator of this program for more information. So that's $30 for it to be easier for you. It can be paid in installments. You can pay the first half and you can pay the second half forward to the end of the program. And so those are some of these things that you may miss, or you miss rather, if you are not part of this, like career training, our research platform, skill advancement. And we know that in this 21st century, bioinformatics is very key for any biological science student. Bioinformatics is an hot key that you cannot do without in this age, because the world is already going to the area of precision. Need to be relevant, have some basic knowledge on bioinformatics, which we are here to offer you. And so we have some bioinformatics tools, we have access to top measures, and we have access to certificate, recommendation letter, global exposure, and more. Please, are we together? Are you following me? Please, if you are, I want you to see some thumbs up there so that we
Yes,
know
we
that
are.
we are together. All right.
We are with you, sir.
Okay. All right. So, and so for us, along for this intern program to be seamless, for you and for us here, we have some policy. We have a no refund policy. Any payment made are not refundable. So you can only register if you have your registration fee show that you are committed to our program. So if you pay and you do not show commitment over the week, there will not be any refund. So it is called a commitment fee, that this payment shows that I am committing to this intern. So we have platform value and conduct. So we want you to respect the opportunity to exploit for your professional growth. So we want you to be disciplined, to be respectful, most especially to be respectful to be guiding you, coordinating you in your doing your internship rather. So you have certificate and report. Only consistent participant who complete all tasks will receive certificate and recommendation. Okay, so we indulge you to be active, participate actively, work submission. Your project will be submitted at the end of the internship. If it doesn't meet journal standard, it will be published on research. It will not be published on research grade. Please let me read that again. What this is trying to tell us is that for your research to be approved, it must meet research standard. If it doesn't meet research standard, it will be rejected. So make sure that and we will guide you on how to do that so that your paper can meet research standard. So each intern is assigned to one cohort. You cannot be in two groups at the same time. You can only be in one cohort and in one group at a time. So you must finish with your cohort and no department or transfer allowed. So at this time, if there is anyone that needs some clarity or question, I will want to entertain question before we go to the next narration. Any question from us or clarification? Is there anyone that has a question? Okay, no question. No question. All right, so I want to wish you congratulations again. Congratulations to all of you, and I want to say it for those coming in, it will be a seamless internship program, the one that you will be able to have the required skill. So at this time, I want to hand over to Ms. Aditola to continue the presentation. Thank you for having me.